I'd like to thank Dr. Dabaja for the invitation here today. So what I thought I'll do today is to discuss with you, give you some recent updates on pivotal trials of CAR T-cell therapy in non-Hodgkin lymphoma, and then move on to current challenges and future strategies for improving efficacy of CAR T-cell therapy in lymphoma. Here are my disclosures. I think most of you are aware that there are at least 3 CD19 CAR T-cell products that have been tested in pivotal trials in non-Hodgkin lymphoma: axicabtagene, cilta-leucel, also known as Axicel, tisagenlecleucel or Tisacel, and lisocabtagene maraleucel or Lisocel. And these cartoons show you the similarities and differences across these 3 products. All of them use the same CD19 antibody clone and have CD3 zeta to provide signal 1 for activation of the T cells, but the co-stimulatory domain differs with the Axicel product using CD28 co-stimulatory domain, whereas the other two use 4-1BB co-stimulatory domain. There are also other minor differences in the hinge and transmembrane region, as well as the gene transfer technology that is used. And this slide shows you the treatment schema for CAR T-cell therapy for most products. The patients initially undergo leukapheresis, and the product is, while the product is being manufactured, they may receive bridging therapy to control their disease. And once the product has been delivered, they receive conditioning therapy, usually cyclophosphamide and fludarabine for about 3 days, followed by CAR T-cell infusion on day 0. And they usually monitor for toxicities, which typically occur within the first 2 weeks after the infusion, and the first tumor assessment is done at day 30. And this table summarizes the 3 pivotal trials: ZUMA-1 testing Axicel, Juliet testing Tisocel, and Transcend trial testing the Lisocel product. Besides the difference in the CAR constructs, there are also difference in the dosages. All 3 trials use cyclophosphamide and fludarabine for conditioning therapy. In addition, the Juliet trial also allowed bendamustine conditioning. And all of them included patients with diffuse large B-cell lymphoma and transformed follicular lymphoma. In addition, PMBCL was included in ZUMA-1 and Transcend trials. Now, if you look at the percentage of patients who actually received the CAR T-cell product versus those who were enrolled on these trials, you'll notice that this was highest on the ZUMA-1 trial at 90% versus 67% on the Juliet and 78% on the Transcend. And part of the reason for this drop-off in these patients after apheresis is because of the rapid progression of the disease, which highlights the need to develop better bridging therapy options for these patients. So the ZUMA-1 showed an overall response rate of 83% and a complete response rate of 58% in large relapsed refractory large B-cell lymphoma, and at a median follow-up of 27 months, 39% of the patients remain progression-free. And what's nice to see here is this plateauing of this PFS curve at around the 6-month time point, with very few relapses beyond that. And at a median follow-up of 39 months, 47% of the patients are still alive. And as compared to the historical control with the SCHOLAR-1 study, which looked at outcomes in 600 patients with refractory large B-cell lymphoma, you can see there's a significant improvement in overall survival from 12% at 2 years versus 50% at 2 years on the ZUMA-1 study, suggesting that this represents a major advance for the management of these patients. And similarly, on the Juliet and the Transcend, we can see very high complete response rate in the 40 to 50% range. And again, we can see a nice plateauing of the PFS curve at around the 6-month time point on both of these studies as well, with very few relapses beyond that. And there are 2 common toxicities that are observed with CAR T-cell therapy: cytokine release syndrome and neurological toxicity. On the ZUMA-1 study, most of the patients developed CRS, but majority of these were grade 1 and grade 2, with only 11% having grade 3 or higher. The lowest toxicity was observed on the Transcend trial, with only 2% having grade 3 or higher CRS. And neurological toxicity is the second most common toxicity observed in these patients. Again, mostly grade 1 and grade 2, with very few grade 3 or higher. But these toxicities are generally reversible with the usage of tocilizumab or corticosteroids for severe toxicities. Now, this slide shows you the ZUMA-2 trial testing KTEX19, which is a product very similar to Axicel, but with slight difference in manufacturing in patients with relapsed or refractory mantle cell lymphoma. Of 74 patients treated on this trial, the best overall response rate was 93%, and complete response rate was 67%. And at median follow-up of 12 months, 61% of the patients remain progression-free. And the safety profile of this product was quite similar to what was observed on the ZUMA-1 study. And based on this pivotal trial, brexacaptagene, or Teleucel or Brexocel in short, was recently FDA approved in the US for relapsed refractory mantle cell lymphoma. And here's the ZUMA-5 study, which is the pivotal trial with Axicel in relapsed refractory indolent non-Hodgkin lymphoma. So the interim analysis from this study was presented at ASCO this year of 140 patients enrolled and treated. The best overall response rate was 93%, and the complete response rate was 80%, with 68% of the patients remain progression-free at 15 months. And what you can see here is that the durability of these responses appear to be better in follicular lymphoma, shown here in the blue curve, versus patients with marginal zone lymphoma, shown here in the teal curve. And this raises the question, is there a role for combination with radiation in marginal zone lymphoma patients, given that this is a highly radiosensitive disease? Now, CD30 also appears to be a promising target for CAR T cell therapy in Hodgkin lymphoma, as well as certain T cell lymphomas. In this recent study that was just published in JCO this year of 41 patients treated on 2 parallel phase 1 trials using a similar product, the best overall response rate was 62% in relapsed refractory Hodgkin lymphoma, and complete response rate was 51%. And the patients received a variety of conditioning therapies, including bendamustine alone or bendaflu or cy/flu conditioning. And the patients who received the bendaflu appeared to have much better CR rate. But unlike what was observed in B-cell non-Hodgkin lymphoma, in Hodgkin lymphoma, the PFS does not appear to be durable, with most patients relapsing by about 1 to 2 years. And you can see here that the patients who receive the fludarabine conditioning appear to do slightly better compared to the non-fludarabine conditioning regimen. But again, this raised the question, is there a role for combination with radiation therapy, given that Hodgkin's is a highly radiosensitive disease? Now, switching back to diffuse large B-cell lymphoma, the important question going forward is, although we see about 40% of these patients have durable responses, the question is, why do 60% of these patients relapse? And if you look at patterns of failure in DLBCL after Axicel, we see that about 20% of these patients have primary resistance. All of them progress by about 1 month. About 80% are responders. And of these, about 60% are complete responses and 20% are partial responses. And the partial responders all progress early, usually within the first 1 to 2 months. And among the complete responders, about 40% have durable CRs, about 10% relapse early within the first 2 to 6 months, and another 10% relapse late beyond 6 months. So there have been attempts to identify potential biomarkers that may be associated with response and durability. And on the ZUMA-1, we looked at a number of different prognostic markers, including the traditional clinical prognostic markers such as age, stage, IPI, refractory status, various product characteristics, as well as tumor characteristics, but none of these were associated with durability or response. But the one parameter that was consistently associated with both response and durability was the degree of CAR T cell expansion post-infusion. And here's data showing you that as compared to the non-responders, the responders have both a higher peak CAR T cell expansion as well as higher CAR AUC levels over the first 28 days. But what's also important to note here is the unique pharmacokinetics associated with CAR T cell therapy compared with traditional therapeutic agents. You can see there's almost a 4 log difference across these 100 patients in the peak levels as well as CAR AUC levels, as opposed to usually less than 1 log difference that are seen with traditional therapeutic agents. And this highlights the need to develop tunable CARs to better control the rate of expansion post infusion and to achieve better clinical efficacy as well as lower the toxicity. Now, another important question in the field is, do we need long-term persistence of these CAR T cells to achieve durability of responses? So we looked at it on the Zuma one in a couple of different ways. Here's data showing you looking at persistence of CAR T cells at the 1 year time point in patients who have ongoing response. And here we found that about 30% of these patients did not have detectable CAR T cells, suggesting that probably long-term persistence may not be needed to maintain durability of responses, at least in lymphomas. And there are similar results were also obtained when we looked at the 2 year time point. But this still raises the question, why do these patients relapse? So one way to think about this is to look at the CD19 status post-infusion. If the CAR T cell therapy is really good and does what it's supposed to do, which is to eliminate every CD19 expressing tumor cell in the body, the most relapsed tumors should be CD19 negative. On the other hand, if you find that most relapsed tumors are CD19 positive, that would mean that we may have to do better at optimizing the CAR T cell therapy efficacy. So I'd like to show you a few examples to give you an idea of the patterns of relapses that we see in these patients, so that can give you some idea on where radiation might play a role. So here's a patient, a 68-year-old male who was refractory to multiple chemotherapies, had extensive skin and lymph node disease at the time of axi-cell infusion. And 1 month after axi-cell infusion, most of the skin lesions disappear, but he had this persistent lesion behind the right knee, which unfortunately progressed by 3 months. Now, when we biopsied this lesion on the shin at baseline, we find that this tumor was CD19 positive. But when we biopsied this lesion that persisted and progressed by 3 months, this was completely CD19 negative, suggesting that there could be heterogeneity in CD19 expression across different sites in these patients. Now, here's a second patient, a 72-year-old male with DLBCL who also had primary refractory disease and had extensive skin, bone, and lymph node disease at the time of treatment with axi-cell. He achieves a complete metabolic response by 1 month, but by 3 months, his disease comes back in the thigh. When we biopsied this thigh lesion at baseline, this was completely CD19 positive, but at relapse at 3 months, this was completely CD19 negative. To me, this suggests that the frequency of the CD19 negative clones in some of these patients at baseline is probably extremely low, probably in the order of 1 out of several billion cells, which highlights the difficulty in trying to develop a sensitive biomarker for these patients, because we currently don't have an assay with that degree of sensitivity. So the question is, how common is CD19 loss? We are, after axi-cell in DLBCL, we see that about a third of these patients may have CD19 loss. And this is thought to be either because of mutations, loss of heterozygosity, or alternative splicing of CD19. The remaining 2/3 of these patients have CD19 positive disease. Now, there's emerging evidence to suggest that the major mechanism of resistance in patients who have CD19 positive relapses is probably impaired T cell fitness. And you can think of impaired T cell fitness at various stages during the course of the patient's treatment of this therapy. The T cells could be dysfunctional at the time of the apheresis because of the disease itself or because of prior therapies that these patients receive. The T cells could become dysfunctional during the manufacturing process, or if the host is not conditioned properly, or when they traffic to the tumor site because of various immune checkpoints in the tumor microenvironment. So in the Zuma 1 study, we looked at the CAR T cell fitness by looking at the doubling time of the CAR T cells during the manufacturing phase. So when we group these patients based on the number of prior lines of therapy into 2 lines, 3, 4, or 5 or more lines of therapy, and looked at the doubling time of the T cells, we found that patients who had 4 to 5 lines of therapy, their doubling time was longer, meaning their T cells are proliferating slower compared to those patients who had 2 to 3 lines of therapy. And the shortest doubling time was seen with healthy donor T cells. And now this doubling time was associated with a lower degree of CAR T cell expansion, especially for the patients who had 5 or more lines of therapy, a lower overall response rate, and a lower durability of that responses. So to me, this suggests that early referral may improve the efficacy of CAR T cell therapy in these patients. Now, the UPenn group went one step earlier and looked at the T cell fitness in the apheresis product. So they did this in CLL patients where the CR rates are much lower at only at about 25% with T-cell compared to DLBCL, where it's in the 40 to 50% range. So they took the CAR T cells in from patients, CLL patients who achieved a CR versus those who did not respond, and injected them into mice with xenografts. And they found that the CAR T cell product derived from the complete responders did much better control of the tumor in these mice compared to the non-responders. And the CAR T cells that performed the best were those that were derived from healthy donor T cells. And they went back and analyzed the apheresis product with RNA-seq as well as CyTOF and found that if the apheresis product had a higher frequency of early memory T cells, which they define as CD27 positive, CD45 auto negative, CD8 positive T cells, then they were able to make a better quality CAR T product, and that associated with a better durable remission in these patients. Now, to me, these data provide a rationale to develop allogeneic CAR T cells or banking T cells when we are healthy. And allogeneic CAR T cells have recently entered clinical trials. Now, the CAR T cell fitness may also be affected if the host is not conditioned properly. So this was tested by the Fred Hutch group, where they gave a 4-1BB CD19 CAR to lymphoma patients, and after 2 different types of conditioning therapy. One group received the Cy/Flu conditioning, and the other group received a non-fludarabine containing conditioning therapy. And you can see here in the red curves, the patients who received the Cy/Flu had a much better CAR T cell expansion, both with respect to CD4s and CD8s, and much better persistence of those CAR T cells. But more importantly, this translated to a better clinical outcome. Even though this was not a randomized study, the patients who received the Cy/Flu had a complete response rate of 50%. But if they received only Cy or Cy-etoposide conditioning, the complete response rate was only 8%. And conditioning therapy is known to act by a number of different ways. It can induce host lymphodepletion of T cell, B, and NK cells, and this can eliminate the homeostatic cytokine sinks and make these homeostatic cytokines such as IL-2, IL-7, IL-15 available for the CAR T cells for better proliferation and expansion. In addition, conditioning therapy could eradicate immunosuppressive cells such as Tregs and MDSCs, or modulate the tumor by decreasing immunosuppressive enzymes such as IDO, or increasing co-stimulatory molecules on tumor cells to make them more susceptible for T cell mediated killing. So there is definitely a role to optimize conditioning therapy, and the question is, can radiation play a role in this space? Now, there's also evidence to suggest that tumor burden is associated with efficacy as well as toxicity in DLBCL with multiple different CAR T products. And this is data from the ZUMA-1 study testing Axicell. And if you divide these patients into 4 quartiles based on the tumor burden as measured on the CT scans by sum of the product of the perpendicular diameters, you can see the patients who have the lowest tumor burden had a much better durability of response at 1 year, with 2/3 of these patients had ongoing response, versus in the highest quartile where only a quarter of these patients had ongoing response. And similarly, the patients who were in the lowest quartile of tumor burden had a much lower grade 3 or higher CRS as well as neurological toxicity compared to the other quartile groups. So this again there is a question: is there a role for radiation in debulking these patients to both to improve efficacy as well as lower toxicity prior to CAR T cell infusion? So here's a third case, a 28-year-old male with high-grade B-cell lymphoma who was refractory to multiple chemotherapies, had exploding disease when he was treated with Axicel. And after 1 month, he achieves a complete remission. We call this a complete remission because this patient still had this tiny speck of FDG avid focus in the pelvic, so which unfortunately progressed by 3 months. And this was a CD19 positive relapse. Now, it's hard to argue that the CAR T cells were not fit in this particular patient since they were able to eradicate most of the tumor. But you have to wonder, why didn't they eradicate this last bit of tumor cells? So one possibility is that there could be immune checkpoints in the tumor microenvironment, which may have impaired CAR T cell activity against these tumor cells, or there could be tumor intrinsic resistance to CAR T mediated killing. So in the Zuma 1 study, we looked at PDL1 expression at progression, and we found that at least 2/3 of these tumors at progression were PDL1 positive. And this provided a rationale to develop combination strategies of CAR T with PD1 or PDL1 blockade post-infusion as a way to optimize CAR T cell efficacy, and those trials are currently ongoing. There's also recent data that suggests that there could be tumor intrinsic resistance to CAR T mediated killing. And this was shown very nicely by Nathan Singh and colleagues from UPenn in a paper that was recently published in Cancer Discovery, where they showed in ALL patients as well as in DLBCL cell lines. Uh, the death receptor gene signature score appear to correlate with susceptibility or resistance. If there is a high death receptor signature score, these patients did much better with CAR T cell therapy compared to an impaired death receptor signaling, in which case there seem to be a resistance to CAR T cell therapy. And this again, there is a question: is there a role for radiation in eradicating this residual disease that is resistant to CAR T mediated killing? But the question is, how do we identify such patients? So one way to identify such patients is to look at the MRD status, and there's emerging data on this. So this was data that was presented at ASH meeting last year, looking at MRD status using the clonoSEQ approach, using the immunoglobulin sequence from the Stanford group, where in DLBCL patients after Axicel therapy, and they found that MRD status at day 28 appeared to strongly associated with durability of response, and it appeared to be a much better predictor than PET scan, PET positivity or negativity. And here you can see that at day 28, if they're MRD negative, about 70% of these patients have long-term durability, but if they're MRD positive at day 28, almost all of them relapse within 6 months. And then this MRD status was also beneficial in patients who had PR or stable disease by PET scan. So, if they're PET positive but negative by MRD status, again, their outcome was comparable to patients who are PET negative. As opposed to MRD positivity, all of these patients relapse within 6 months. Now, Michael Green from our group at MD Anderson actually found that you could actually look at MRD status as early as day 7. In this paper that was just published in Nature Medicine, he found using the CAP-seq approach in cell-free DNA sequencing, if the patients have a more than 5-fold reduction in variant allele frequency as measured by cell-free DNA sequencing at day 7 after Axicel therapy, 75% of the patients have durable remissions, as opposed to if they had less than 5-fold reduction in MRD status, then all of these patients relapse by 3 months. Now, these are small numbers of patients, as is now being confirmed in a larger group of patients, but you can see here, these patients tend to have rapid molecular response by day 7, and this may potentially be used to develop consolidation strategies. And one question is, can this help in identifying patients who may benefit from radiation post-infusion? So to summarize, in order to improve efficacy of CAR T cell therapy in B cell malignancies in the patients who are developing CD19 negative disease, one strategy is to target multiple antigens, and these are ongoing, targeting CD20, CD22, BAF receptor, CD79B. And in the patients who develop CD19 positive relapses, we have to we have to think of strategies to improve T cell fitness. And again, we can think about this at various stages during the course of this treatment. At the stage of the apheresis, we can think about an allogenic source of CAR T cells, or select specific T cell subsets. There's also evidence suggesting that ibrutinib in CLL patients giving before apheresis may improve the T cell fitness. One can alter the CAR T cell product itself or the manufacturing process, but one may also need to think about strategies to improve the conditioning therapy. And here's where radiation might play a role, both for debulking as well as optimizing the conditioning before CAR T cell infusion. And one may also need to think about post-infusion conditioning or treatment of resistant disease. And here's where I think radiation can play a role as well. So to summarize, autologous anti-CD19 CAR T cell therapy induces durable remissions in approximately 40% of patients with relapsed or refractory large B-cell lymphoma. Although longer follow-up is needed, this therapy appears to have promising activity in mantle cell lymphoma as well as indolent non-Hodgkin lymphoma. CRS and neurological toxicity are the major toxicities, but generally reversible. And targeting multiple antigens and strategies to improve T cell fitness may improve efficacy of CAR T cell therapy. CD30 appears to be a safe and promising target for CAR T cell therapy in Hodgkin lymphoma and certain T cell lymphomas. And finally, radiation may have a role in reducing tumor bulk and optimizing conditioning pre-infusion or post-infusion conditioning or therapy of resistant disease guided by MRD status to improve CAR T outcomes. So with that, I'd like to thank you for your attention and happy to take any questions.
3, 2, 1.
All right, well, thanks a lot for inviting me. I'm really excited to be here and excited to discuss these topics for introducing radiation to CAR T cells with everyone. My name is Carl DeSelm, and I'll start out with a little bit of background and my disclosures. I have not accepted any gifts or financial contributions from industry or companies. And I'm going to talk a little bit about radiation with CAR T cells and provide a couple of examples from a couple of patients that were treated at Memorial Sloan Kettering while I was there, and then some follow-up laboratory experiments based on the results of those patients. And this is going to be some hypothesis generating data that perhaps might inform some more definitive studies in the future. So I'd like to point out that we all are aware that radiation may affect the immune response, and the abscopal response has become a popular concept in radiation oncology. And the proposed mechanism for this response is that radiation causes tumor death and the release of DAMPs and PAMPs, which attract antigen-presenting cells. Those antigen-presenting cells take up dead tumor and then migrate to the lymph node, where they provide stimulation and co-stimulation to endogenous T cells. Those endogenous T cells can then circulate in a more activated state and encounter additional tumor cells and kill them more effectively. And in addition, radiation enhances the expression of MHC on tumors that have been directly irradiated, which can improve the expression of tumor antigens and further activate T cells and further improve tumor killing. Now, I wanted to point out that none of these mechanisms should theoretically apply to CAR T cells, because CAR T cells obtain their stimulation and their co-stimulation through the CAR and not through antigen-presenting cells. And they don't kill through their own T cell receptor. So if radiation does affect the biology of CAR T cells, then it's likely in a completely different mechanism than endogenous T cells. So it leaves us to wonder, does radiation affect the biology of the CAR T cell response? And whether or not it affects the biology, is there a role for radiation with CAR T cells? So the first patient I'm going to talk about is a DLBCL patient who was diagnosed in 2015, had a number of different chemotherapy regimens and progressed including stem cell transplant. This is the baseline PET scan. MIP, you can see there's a lot of different sites of disease, including disease within and outside of the bone. And one thing that was interesting about this patient is that he had a very low level of CD19 and low to absent level of CD19, most people would say. So this control patient, you can see on the right, CD19 is expressed on 100% of the tumor cells. CD10 is another tumor cell marker. And then the patient that I'm showing here had CD19 loss on a lot of the cells at baseline. And we confirm this with a biopsy. This is a immunohistochemistry stain of CD19 on the tumor biopsy. And you can see there are a number of tumor cells that are still positive, but there are also a lot of tumor cells that are negative at this point. So he was still treated with CD19 CAR T cells on a clinical trial. And before giving him the CAR T cells, we decided to irradiate one of his legs. He had some symptomatic disease in the skin of his legs. So we irradiated one of these with 400 times 5 and then proceeded with delivery of CAR T cells. This was done after the leukapheresis, but before the CAR T cells were delivered. And 1 month post-treatment, he had an excellent response, especially given the expression levels of his CD19. He had what was read as a complete response by PET scan systemically, both within and without the radiation field. But then a month later, he recurred systemically in multiple places, multiple old sites of disease as well as new sites of disease, but he didn't recur within the radiation field. And this 2-month scan is shown here. Here's a 6-month scan. He remained disease-free within the radiation field. And until he eventually died, he never recurred within the radiation field, although he recurred multiple areas outside of the field. So the other leg, for example, you can see the disease burden distribution. Many of those sites recurred, and then he had many new sites within his other leg, but he didn't develop new sites within the irradiated field. So we were intrigued by this phenomenon. First of all, why did he have a lasting complete response within the radiated field? He got 4 gray times 5, which often does not cause a lasting complete response in an aggressive DLBCL like this. And also, why did no new lesions develop within the field, even though they developed systemically? So we started to try to answer some of these questions in the laboratory. And the first thing we wanted to know was, does sublethally irradiating a tumor affect the ability of the CAR T cell to kill the tumor? So we took a human leukemia cell line, NALM6, which is derived from a 19-year-old with ALL. And we gave those cells 1.8 gray of radiation. And then 2 days later, we selected the live cells. So these are the cells that are resistant to this 1.8 gray, relatively low dose of radiation. And then we incubated those with CAR T cells, and we compared them to the same number of live cells that had not previously been exposed to radiation. And then we just assessed the amount of killing that occurred over 24 hours. And you can see that at every ratio of CAR T cells to tumor, so effector to target, this is 1:1, 1:2, down to 1 CAR T cell per 32 tumor cells. You can see that the amount of killing is higher for all levels in the tumor that had previously been exposed to this low dose of radiation. So we wondered, does this sensitization occur in mice? So we decided to test a model where we injected these ALL tumor cells into immunodeficient NSG mice. And then 4 days later, after the tumors had been established, we gave the mice 1.8 gray of radiation. And then we treated them with a very low dose of CAR T cells. So this is only 10,000 CAR T cells. Generally, we would treat these mice with around 500,000 CAR T cells to eliminate the tumor. So a much lower dose. And this was based on the in vitro results that there's a significant amount of sensitization that occurs in vitro. So if we expected the same thing to occur in vivo, then 10,000 CAR T cells may in fact work. Now you can see these mice, this is the bioluminescence imaging every week. The radiation plus 10,000 control CAR T cells, which are targeting an irrelevant tumor antigen that's not expressed in these mice, start to progress at 3 weeks. The radiation alone mice, so these are ones that only got the 1.8 gray of radiation, progress after 3 weeks. The 10,000 CAR T cells alone mice look like this, and the ones that had no treatment also progress at a similar rate. Now, the mice that had pre-irradiated systemically with 1.8 gray exhibited a very different response, and you can see they never really developed a large burden of disease, and in fact, until they basically died of other causes, most generally graft versus host disease, but they never developed disease, and they exhibited complete tumor elimination. So this was more dramatic than I was expecting. And you can see the survival curves here are very different. And the tumor burden, you can see in the mice that had 1.8 gray plus 10,000 CAR T cells, the imaging did detect a significant level of disease at week 2, which is this week up here. And then that disease eventually regressed by week 3 and then never came back. So we wondered what was going on. You know, is this just a debulking phenomenon? Maybe the small dose of radiation eliminated enough tumor cells that the small number of CAR T cells were able to kill the remaining cells, and it's just a simple debulking phenomenon? Or is there something more interesting that's maybe affecting the biology and maybe sensitizing tumor cells to CAR T cell killing? Because it did seem to be more of a synergistic effect. So we did RNA sequencing of the tumor after low-dose radiation, and we found that a number of pathways are both enhanced or reduced by low dose, but some of the most significant ones are apoptosis pathways, and among those, the trail apoptosis pathway, which is the death receptor pathway, was very significantly elevated after low-dose radiation. So I learned more about trail. Trail is a molecule that's secreted by a number of cells or expressed by a number of cells, including T cells. And we found that CAR T cells actually express a much larger amount of trail after they are activated by their target. So these are days post-stimulation of CAR T cells on their target. And you can see after 2 and 3 days, the mRNA levels start to go up for trail in the CAR T cells, and the amount of secreted protein, which is measured by ELISA in the supernatant, starts to go up at 3 days and continues to be high as far out as we tested, which was day 11. So the CAR T cells start to secrete more trail as they become activated. The low-dose radiation increases the amount of trail apoptotic signaling molecules in the tumor, which makes them more sensitive to trail. So this is possibly a mechanism of synergy. And I think it's interesting, I'm going to point out a related study that was recently performed by Nathan Singh, who coincidentally I now work with at WashU, but who did this study while he was at Penn. And they basically did a CRISPR screen where they infected this ALL tumor cell line with a library of guide RNAs so that every single gene in the genome was knocked out in a single cell only, and then they incubated these cells with CAR T cells. And after that, they looked at the genes that had been enriched. So these are genes whose loss would enable resistance to CAR T cells. And they also looked at the genes that were depleted. So these are genes that would sensitize to CAR T cells. And they also found that resistant tumor cells frequently had impaired apoptotic signaling. And there are a number of different apoptosis pathways, but notably they found that all of the guide targets associated with the apoptosis gene set belong to the death receptor-driven apoptotic signaling pathway, suggesting that death receptor signaling is a key regulator of primary resistance in CAR T cells in ALL. Now, this was all in vitro, but they went on to look at patients who they had banked samples before CAR T-cell treatment. These are pediatric patients treated with Tisacel. And they looked at those who had a complete response versus those who did not have a response. And they found also that the death receptor signaling pathway was significantly different. So they were able to come up with a score based on high death receptor signaling pathway baseline expression versus low expression. And overall, these patients did quite well, but those who did not do well had a low death receptor signaling score at baseline. And these patients did significantly worse. So going back to our data, we had found that the T-cell, the CAR T-cell, expresses trail at an enhanced level after it finds its target. Low dose radiation has a pretty dramatic effect on the baseline death receptor signaling pathway. I went back and just mapped out all of the known molecules in the death receptor signaling pathway, and then show here based on color whether these molecules are increased. So the more red, the more increased they are. The very red ones are threefold increased. The more green, the more decreased they are. So the very green ones are fivefold decreased. And you can see that tumor supportive inflammation molecules are decreased by low dose radiation. Survival and migration molecules are decreased by low dose radiation. Molecules that internalize the death receptor are also decreased by low dose radiation. So you have enhanced and maintained expression of the death receptor itself. And then the downstream necroptosis and apoptosis pathways in the death receptor pathway are largely increased by low dose radiation. In here, the molecules that are decreased are those that inhibit the death receptor apoptosis pathway. So it's tempting to speculate whether if we took these patients who had a low death receptor signaling pathway at baseline, and we gave them a low dose radiation, which significantly enhances their death receptor signaling pathway expression, if we could potentially move this curve up to here. Now, another question I mentioned was the patient who we treated to the leg who never had a recurrence within the leg. We wondered, does radiation prevent tumor reseeding in the area by perhaps changing the tumor microenvironment or changing the microvasculature? So we did a similar model in mice. We took leukemia cells and we took immunodeficient mice, injected them with the tumor, and then we just irradiated their one leg as we did with this patient. And then we treated them with CAR T cells. And we found that the week following radiation, they did have a reduced number of tumors developing in the irradiated leg. But eventually this caught up and seemed to be no different at all in both sides of the leg. So my conclusion from this very small experiment was that radiation did not necessarily prevent tumor reseeding in this area, but perhaps just helped to completely eliminate the tumor within the radiation field in the beginning after CAR T-cell treatment. Now, one major caveat to this experiment is that these are immunodeficient mice, and the situation may be completely different in mice or patients with an intact immune system. Another very important question is, what is the best timing for radiation in CAR T cells? Now, this is a difficult question to answer in mice especially, and one of the reasons is that while we wait to give the CAR T cells in patients, we generally treat them with other therapies to keep their tumor burden down, and that's a little bit hard to do in mice. But in any case, we tried to model this question by injecting tumor into the mice and then treating them with radiation 4 days later after the tumor had established, and then waiting to give the CAR T cells either that day of radiation or the day after, within 24 hours basically, or waiting 1 week and giving the CAR T cells a week after radiation. Now, when we looked at these patients who are these mouse patients who were treated the day after radiation with CAR T cells, in the first 3 weeks, they all looked very similar. These are 1 day after, these are 7 days after. And at week 3, they all looked very bad. They looked like they were all nearly about to die. But we came back the next week, and 2 of these mice who had been treated the day after with CAR T cells actually recovered. And the following week, they looked fantastic, and they eventually became extremely healthy and lived a long life. So in this experiment, anyway, radiation that was preceded very quickly by CAR T cells had a significant survival advantage in mice relative to those that were treated later. And again, this is a very low dose of CAR T cells, very low dose of radiation. And you can see the tumor burden was actually completely unaffected over the first 3 weeks, but the only difference was that some of these mice eventually recovered in the mice that were treated early with CAR T cells. So some of the conclusions from the first patient and the first experiments that were performed after this is that radiation, even at a sub-definitive dose, appears to sensitize at least some tumor types to CAR T cell killing. You have induction of the death receptor pathway, which appears to be an important pathway for killing by CAR T cells. Now, I think it's important to recognize any potential caveats. One that I can think of here is that these tumors are not heavily pretreated. They appear to be highly sensitive still to both chemotherapy and radiation, and the results may be a little bit different in patients who do have much more resistant tumors. Tumor control within the irradiated field may be due more to complete tumor elimination within the field rather than prevention of tumor reseeding, at least in immunodeficient mice. And administering CAR T cells sooner rather than later after radiation is beneficial in mice anyway. Now, what about giving radiation after CAR T cells? This is something that I think many people avoid very reasonably because there's the consideration that radiation will likely kill off a lot of the CAR T cells if we are directly irradiating the cells anyway. But let me tell you about a patient who had radiation after his CAR T cells. So this is a 63-year-old woman who was diagnosed with multiple myeloma with high-risk cytogenetics. She had a number of different previous lines of therapy, including stem cell transplant, and then enrolled on the BCMA clinical trial at MSK that had a 4-1BB co-stimulatory domain. This is her baseline PET scan. You can see she had extensive disease both within the bone and outside of the bone. Her M spike was 2.26 at baseline, and her baseline bone marrow biopsy showed 95% plasma cell infiltration. So she really had a large disease burden. She received Cy/Flu, lymphodepleting chemo, followed by a single infusion of CAR T cells. And then shortly after her CAR T cell therapy, she exhibited signs of lower extremity weakness and confusion. She was awake and alert, but her mental status was impaired. She had decreased short-term recall. She couldn't recall any objects out of 3, and she had impaired attention and calculation. She couldn't spell world backwards. MRI was obtained of her spine and her brain, and she had cord compression. You can see there's gross disease within her spinal canal. She also had these pleural lesions nearby that were progressing. In her brain, she had a large epidural mass that was causing midline shift. And 5 days after CAR T cells were given, the M spike had increased from her baseline to 2.83. And her neurological symptoms began to resolve at this point, but the lesion compressing her thoracic spinal cord on imaging and her increasing multiple myeloma markers were concerning for disease progression and cord compression. So she received high-dose steroids at this point. She got 10 milligrams every 12 hours for 13 days. And she also received palliative radiation to the thoracic spine, as well as whole brain radiation. And this was delivered between day 6 and 20 post-CAR T cells. She got 20 gray and 5 fractions to each site. And this is what the fields look like. This is the thoracic spine, and this is the whole brain field. And her MRI at 4 weeks showed a nice disease response at the sites of radiation. So she no longer had the disease within the spinal canal. Her pleural disease was greatly decreased, and the disease that had been compressing her brain had substantially diminished. And then after radiation, she also started to respond systemically. So a PET scan done at 8 weeks after CAR T cells showed a complete radiographic response, including the innumerable sites outside of the radiation field. And this response persisted through 9 months post-therapy. Again, this is the baseline, and then this is the 8-week post-therapy scan. Very nice complete response. And this is a graph of her M spike. So you can see how it increased in the first 5 days after CAR T cells were given, and then radiation was started. It started to level off and it started to decrease, and then continued to decrease out to week 51, at which point it was undetectable. Looking at her T cell receptor clones, interestingly, the number of low expressing T cell receptor clones remained low after CAR T cells, but then radiation was started. And then after radiation, these low expressing TCR clones dramatically increased. So the TCR diversity, as I mentioned, was stable in the beginning, but then a spike of expansion was noted 8 days after radiation finished. And these newly expanded clones made up more than 30% of the T cell repertoire at 4 weeks. And many of these persisted through 100 weeks post-CAR T cell infusion. Notably, there was no expansion of TCR clones that were highly expressed at baseline. So it was all of these just low expressed TCR clones. Now, CAR T cells represented 10% of the patient's T cells at 5 weeks. You can see this is a flow plot of the CAR modified marker and then CD8. So you can see the majority of these CAR T cells were CD8 positive, and these represented 10% of the cells at 5 weeks. Looking at different cytokine levels, IL-6, which is in blue, peaked after radiation was ended, and CRP also peaked after radiation therapy. Ferritin and D-dimers had slowly risen throughout radiation and then began to decline. They also preceded IL-6 and CRP. Temperature did not correlate particularly well with radiation. There was also a small peak after radiation ended, though. So some conclusions from the second patient. High-dose steroids, first of all, did not mitigate the efficacy of CAR T cell therapy, and she did get a very high dose for 13 days. The timing of the CRS, like clinical signs and the inflammatory markers, such as IL-6, coincided with expansion of new TCR clones after radiation therapy, which also correlated with M spike reduction and clinical response, supporting potentially the hypothesis of synergy between radiation and CAR T cell therapy, even when radiation is given after CAR T cell therapy has been given. So I'd like to thank you all and thank the members of my lab in the Center for Human Immunology and Immunotherapy here at WashU. Also John DiPerzio, who runs the CAR T cell therapy department at WashU. Bob Schreiber, who runs the Center for Human Immunology and Immunotherapy, which houses my lab. And the group at Memorial Sloan Kettering, especially Michelle Sadelain and Rainier Brenchens, Eric Smith, who ran the BCMA trial. So happy to open up for any questions. Thanks a lot.